Hello, everyone, and welcome to today's webinar, Basic Introduction to the Science of Cell Sorting. I'm your host, Stephen McClellan. I'm currently the Chief of the Flow Cytometry SRL at the University of South Alabama Mitchell Cancer Institute. This webinar today will be presented by Matthew Goff. Matthew attended Virginia Tech, where he started to learn about flow cytometry, and later took a job as a core manager at the Eastern Virginia Medical School. After leaving EVMS for industry, Matthew stayed close to flow cytometry, supporting both research and industrial scientists in our field, both reagents and software and hardware products. In 2021, he became a commercial product manager at Beckman Coulter Life Sciences and is focused on sustaining the Cytoflex program and launching new products. Matthew is currently the Senior Product Manager of Flow Cytometry at Beckman Coulter. Please help me welcome Matthew.
Thank you for joining the Cyto University presentation, a Day 1 introduction to the science of cell sorting. During this presentation, we will begin with a high-level overview of why and how we isolate cells before narrowing our focus to electrostatic cell sorting. After we establish an understanding of how cell sorting works, we will discuss what factors contribute to quality outcomes when using a cell sorter. As we begin, notice that we're starting with the term isolation. Cell sorting is one of many isolation techniques available to researchers and has some unique properties that can make it more or less desirable depending on the intended outcome. Before we get to sorting, we should first learn why someone would want to isolate cells. Several examples can be seen here. Researchers may employ isolation techniques for single cells to create single clone cultures following a CRISPR process. Others might want to characterize those cells immediately without additional culturing using metabolomics, proteomics, or genomics. A cell therapy researcher might use isolation methods to collect CAR T cells from a PBMC fraction. In general, the need for cell isolation is predicated upon a need to collect a single cell or cell population from a mixed population of cells in suspension. While this is not an exhaustive list of options for cell isolation, it does represent some of the more popular methods. Starting with our non-mechanical methods, we see panning and magnetic bead. Moving down, we get to our first instrument-dependent methods, wherein cells are sorted singly with either piezo-based methods involving the movement of a trapdoor or other gating mechanism. For large cells, researchers will rely upon cytometry-based detection paired to a time-wise air-propelled sorting that pushes the cell out of a single file stream. Lastly, we arrive at electrostatic cell sorting, though we often, if erroneously, omit the term electrostatic. In this class of instrument, there are two main modalities for detection: sense in air, which uses a detection system after the stream exits the flow cell, and sense in cuvette, which interrogates the cells inside of the flow cell. While both have some advantages and disadvantages, modern instruments are remarkably similar aside from the interrogation, and so for the purposes of this presentation, they will be shown together separately, focusing on their commonalities. In the table below, we'll show a comparison of cell isolation techniques. Each of the cell isolation methods listed below has distinct advantages. Because cells are processed in 2 or 3 dimensions with non-mechanical methods, these are the fastest available. They're also the best for working with a large number of target cells. Think millions from billions. However, these isolation methods typically only allow for targeting a single cell or group of cells and doesn't allow for complex decision making. Mechanical methods are typically best suited for special purpose cell isolation. This can include factors like large cell size or especially fragile cells that might be harmed or significantly altered by a more stressful sorting process. In the past decade, there has been a significant amount of development in the technology needed to advance microfluidic sorting. Newer instruments and techniques have increased speed, reducing one of the key differences between microfluidic and electrostatic sorting. Electrostatic sorting offers medium throughput compared to other techniques. While it can be an order of magnitude slower than bead-based cell isolation, it is still faster than most mechanical methods. The key advantage of electrostatic cell sorting over other techniques is target specificity. Because flow cytometry is the detection method used ahead of the cell sorter for target identification, users are able to create complex decisions and conditions for their target cells. Our first example of a cell isolation technique is known as cell panning. Cell panning is pretty straightforward and is a great place for us to start. In order to pan for cells, capture antibody must be annealed to a plate. Cells are then captured by the antibody annealed to the plate and either retained as a negative isolation step or later eluted in order to sort those specific cells that were targeted by the antibodies on the plate. Magnetic bead cell isolation offers users a variety of methods for isolating cells of interest. For simplicity, we'll focus on positive selection and negative selection. In positive selection, the target cell is labeled with antibody tagged with a magnetic bead. Once placed inside of a magnetic apparatus, the labeled cell suspension is passaged through the column where the tagged cells are retained and the untagged or non-target cells are washed out. Once the column is removed from the magnetic apparatus, the target cells can be flushed out in a process called elution. Negative selection, then, is the exact opposite of positive selection. Many researchers commonly use negative selection as an upstream method to enrich cell populations ahead of electrostatic cell sorting. This is done by tagging cells that are unwanted to capture them in a column and remove them from the cell suspension, thus allowing for the target cells to flow through and be used in subsequent assays. Depending on the abundance and the robustness of the cells in question, a researcher might combine multiple positive and negative selection steps ahead of or even in place of more specific sorting methods. One of the key advantages we haven't discussed is that magnetic separation can be used aseptically with less effort than other isolation techniques. At this point, we'll transition away from strictly antibody-mediated methods for cell isolation into instrument and antibody-mediated methods for cell sorting. What does sorting do? Well, in the words of Washington Post reporter David Brown, finding and sorting a few cells from a great mass of them is sometimes likened to finding a needle in a haystack, but the job is more like watching a parade of 1,000 undertakers, spotting the 2 who are wearing yellow ties and the 3 wearing pink ones, and then getting them out of line without making everyone else lose a step. First, we wrap our minds around the elements of a cell sorter and what a cell sorter is, starting with the notion that all fluorescent sorters are first cytometers. They require an input sample and flow cytometric analysis. After the cytometry step is completed, the sorting hardware takes over. To that end, not all fluorescent sorters are electrostatic. Some of them are mechanical. On the lower left, we see a gravity mechanical sorting apparatus associated with a legacy analytical flow cytometer. In the middle, we see what we previously described as an air sorting apparatus, generally used for large particles. And on the lower right, we see a schematic representation of a microfluidic sorting chip used in a modern microfluidic sorting instrument. It's important to know where we've come from before we discuss what we're doing. So with that, I'd like to share some key events in the early development of cell sorters. Notably, in 1949, Wallace Coulter develops the Coulter method, which is based on the science of passing a cell through a charged orifice and measuring the impedance created by that cell to determine its size. In 1965, Mac Fulwiler develops the first electrostatic cell sorter, and he does this by pairing the technology invented by Wallace Coulter to an inkjet printer invented by Richard Sweet. In the 1960s and 70s, Leonard Herzenberg contributes to the development of the fluorescence-activated cell sorter, while simultaneously developing the monoclonal antibodies and fluorochromes we use today to identify cells. And now we've arrived at our first look at the functional description of how an electrostatic cell sorter works. We'll talk about this in 5 distinct steps, and you can see on the left that we show the sensing cuvette system compared to the sense air system that we discussed earlier. Our first step involves optical interrogation and light collection. This is where the two methods are the least similar. You can see on the left with the sense cell or sensing cuvette system that the optical interrogation occurs inside of the flow cell, while on the right, the optical interrogation occurs below the flow cell or after the stream has exited the nozzle. The second step then is stream partitioning into droplets. This is done by applying electricity to a piezo crystal embedded inside of the nozzle and shaking it in a controlled fashion. The third step, stream and droplet charging as the target particle passes through the breakoff. This is done in a time-wise fashion so that the charge is applied to the stream just as the droplet is getting ready to break off from the stream. 4. Droplet deflection to an electrostatic field. Each droplet is charged according to the decision made in the flow cytometer part of the system to allow it to descend through the electrostatic field and then deflect into the appropriate collection device. Step 5 then is the uncharged droplets. These typically pass into waste or are collected by another apparatus for downstream processing. The critical takeaway from this is that we understand that cells are detected, but droplets containing the cells are sorted. It's also an important note that droplets are often large enough to contain more than one cell. Hang on to that piece of information as it will become very important as we move forward. While the 5-step description in the previous slide summarizes the operation of a cell sorter, we need to dive into the subsystems to really understand what is happening behind the scenes. We'll start with the charge. In the picture on the left, we see the graphical representation of a charge called out as a variable, traveling down the stream and arriving at the last attached droplet. The charge applied to the stream corresponds to the decision during detection and allows the droplet containing a target cell to deflect within the electrostatic field into a collection tube, plate, or other device. On the right, we zoom in on this and focus on the droplet creation. We see the same charge traveling down the stream arrive at the last attached droplet, allowing its value to be associated with the sort decision. Users will see terms like amplitude or force of vibration and frequency or time period of vibration associated with droplet maintenance in the sorter control panel. While the frequency of droplet formation does not change, the amplitude may vary to maintain the distance between the point of interrogation and the last attached droplet. This stability is critical for our next task, which is timing. The concept of drop delay is how operators deal with time, distance, and uncertainty within a cell sorter. How long does the instrument wait between event detection and when a charge is sent to the last attached droplet? Drop delay is the amount of time it takes a cell or particle to travel from the point of interrogation, seen in the illustrations on the left and the right as the red circle, to the last attached droplet, represented by the yellow square. As fluidic pressure and droplet frequency inputs are constant and amplitude is variated to maintain the vertical position of the last attached droplet, we're able to use distance to describe timing. The unit of measure for this distance timing is a droplet period, or more simply, drops. For example, if someone were to describe the drop delay as a value of 35, what they mean is that there is an equivalence of 35 sortable droplets between the point of interrogation and the last attached droplet. While we could choose to describe timing in fractions of a second, it would require a rather clunky display of values behind the decimal point, and we would lose the relationship back to the work being done, which is the sorting of cells within droplets. While the inputs for fluidic pressure and droplet frequency are constant, and the amplitude is adjusted fractionally in an ongoing fashion to maintain a constant timing via constant distance, we have to deal with some uncertainty and factor this uncertainty into sort decisions. Uncertainty in droplet formation and position is accounted for in each sort decision based upon the position of a cell within a droplet. Now that we've defined the drop delay, it's important for us to explore methods for determining that drop delay. Prior to the advent of modern instruments with integrated cameras paired to computerized powerful image processing, users would test sort 100 drops onto a microscope slide and count the number of particles in each spot using a fluorescent microscope. Working back and forth across drop values and reducing the range on each subsequent test run would allow an experienced operator to determine the drop delay with a great deal of precision and accuracy. However, this method is costly in terms of time and requires a great deal of practice. Thankfully, this is now an automated process on modern instruments. The instruments calculate the droplet period by observing droplet position or detection of particles in a test scenario during instrument setup. Test scenarios are created by changing the timing described as drops and determining which scenario creates the best sort based upon the presence or absence of particles in the side stream or waste stream, respectively. A third method also exists, wherein a camera attached to an elevator with precise vertical positional control can be used to view droplet formation, thus estimating timing without the use of fluorescent particles. Any and all of these methods are entirely valid and well practiced for the delivery of precise and accurate drop delay results. Once the cytometer within a cell sorter has determined that a cell meets the requirements to be sorted, it communicates the sort decision back to the sorting hardware. To determine whether or not a sort will happen, the sorting hardware must know where the cell is within the stream. In the graphic, the time periods are denoted as future droplets. These horizontal lines represent the beginning and end of each droplet, allowing the sorting hardware to determine the spatial position of each cell. While the ideal position for each cell is in the center of the droplet, this cannot be the case in every scenario. To address this variance, the instrument has programmed tolerances for uncertainty. I will illustrate the practical application of this in the next slide. In the illustration below, we see representative examples of each sorting mode. While variants of each mode exist across the different cell sorter manufacturers, we can use these examples for a simplified discussion. Before we discuss the sorting modes, we should discuss what target cells, non-target cells, and subthreshold particles are and how they each affect the sorting modes. Target cells are those identified in the analytical step and are selected for sorting by an operator. Their opposite is a non-target cell, or those cells intentionally excluded from sorting. Subthreshold particles are a bit trickier. Because cell sorters have a limited amount of computing power, it can be advantageous to reduce the burden of analysis and sorting hardware by raising the threshold closer to the cells of interest, and thus reducing the number of events a sorter must analyze. The cells, particles, and other debris that falls below the threshold cannot be seen by the instrument. The instrument can't exclude or include cells it cannot interrogate, so these may or may not be included in each droplet. Each mode represents a different set of rules for sorting focused on inclusive or exclusive decisions. Purity mode is selected by operators who want to sort the target cell to the exclusion of non-target cells. Enrichment mode is selected by operators who want to sort their target cell, but do not want to lose a cell because of the presence of a non-target cell. Single mode is selected when operators want one target cell per drop, which means that the cell must be outside the uncertainty range for a particular drop. The uncertainty exists to tell the sorter when it needs to account for the preceding or following droplet when making a sort decision. For purity and enrichment modes, a target cell in the uncertainty zone will require that both droplets are sorted so that the particle isn't missed, as long as all the other conditions are also met. In the first scenario, the target cell is in the center of the droplet. All three sorting modes would allow this droplet to be sorted based on the cell's position. In the second scenario, the target cell has moved into uncertainty, necessitating a two-droplet sort, which isn't allowed in single sorting mode. In the third scenario, the target is once again in the center of the droplet, but the instrument now sees a non-target cell. However, this non-target cell is outside of the uncertainty and does not affect the sort decision. All three modes will sort this target. In the fourth scenario, the non-target is moved to the following droplet above our target containing droplet and is in the area of uncertainty. Therefore, this sort would only happen in enrichment mode. The fifth scenario has the non-target cell in the same droplet as the target cell. Once again, this droplet is only sorted in enrichment mode. In our sixth and last scenario, the non-target cell is preceding the droplet, but outside of the uncertainty associated with the droplet containing our target. This meets the conditions for a purity or enrichment sort decision, but fails to meet the conditions for single sort mode, as the target is in the uncertainty zone of the droplet. Every instrument will have its own implementation of sorting modes. It's important to understand what each does and how it affects your downstream collection strategy. Now that we have talked about sort decisions and sorting modes, it's time to discuss the need for speed. The speed of our sort will be dictated by our analytical speed paired with the percentage of our target cells in our sample tube. Our sorting speed is further reduced by a number of other factors. Software aborts are the intentional exclusion of cells from analysis or sorting due to their proximity to one another during acquisition. If a cell arrives close enough in time to another cell, the processing computer inside of the sorter may exclude it from analysis based upon the programmed conditions. A similar decision is made if the cells are further separated but still spatially in conflict according to the sort mode rules being applied to the target population. As we increase the flow of cells into the system, we also encounter another problem: hardware aborts. Simply put, cell sorting systems have some amount of dead time in their electronics, wherein a sample cannot be analyzed. Cells encountered during this time period must be discarded. In the table below, we see an example of a sorter analyzing a sample at 10,000 events per second. Without taking into account any form of cell loss due to aborts, it would take this sorter 100 seconds to sort 1,000 cells, with a 0.1% target cell percentage relative to the sample. By utilizing the enrichment strategies available with either the instrument being run in enrichment sorting mode or using a negative selection strategy, with magnetic bead isolation, an operator can greatly reduce the amount of time required for each sort. When the task is depositing a single cell repeatedly into a 96-well plate, this may not be important. But for bulk sorting into tubes, where a minimum volume of cells must be collected, it can be the critical difference between a 28-hour sort and a 2.8-hour sort, or a 17-minute sort. Once we account for aborts, we must increase our time estimates anywhere from 10 to 50% or more. While discussing speed, an important observed phenomenon in cell sorting is colloquially referred to as the 1/4 principle. When a cell sorter is operated with an event rate in excess of 1/4 of its droplet production frequency, there will be a precipitous drop-off in efficiency. This means that, unless efficient collection of your sample isn't a critical outcome, operating the system above 1/4 of the event per second frequency ratio will lead to a less desirable outcome. In practical terms, this means an operator would not set an instrument creating 40,000 droplets per second above an acquisition rate of 10,000 events per second. In discussion, most operators will try to describe cell sorter performance using a combination of 4 terms: efficiency, purity, recovery, and yield. While the definitions may vary from author to author, what each is trying to describe is reasonably aligned. Efficiency describes the software calculation of sorted cells versus aborted cells. Unfortunately, the instrument cannot take hardware aborts into account, as these are caused by mechanical limitations. Purity comes from the reacquisition of a sorted sample. It describes the amount of target versus non-target cells sorted. Experienced operators know that purity can still be achieved on a sorter with an inaccurate drop delay, which limits the utility of this measurement as a proxy for sound instrument operation. Operators and users must focus on quantitative measurements for quality operation. Recovery and yield, then, are the most critical elements and also have the most variability in definition between authors. Recovery describes the number of target cells in the sorted sample versus the expected number of cells based upon the efficiency reported by the cell sorter. For precision, an operator must sacrifice an aliquot of the sorted sample for volumetric counting as part of reanalysis or separately on a cell counter if the cytometer cannot accommodate this measurement. Yield separately describes a holistic outcome that takes into account all sources of cell loss. It is calculated in much the same way as recovery, but makes the comparison to the input sample rather than the efficiency reported by the instrument. Yield determines how much sample must be input into the instrument to collect the desired sort. As we look to the illustration, we see that each element of our flow sorting workflow gives us a body of information. Starting with our input sample, we see that we're able to obtain information about cell count or density and sample viability, which we describe separately from population viability. In the cell sorter, we can learn about the efficiency of the sort, target cell abundance, sort speed in events per second, sample pressure, population viability, and doublet identification. In the sorted sample, we're able to measure things like purity, population viability again, and cell count or density. Having described the operation of the cell sorter now in great detail, we transition away from the instrument and move towards the cells. Thus, we must know thy cells. Before starting any sort, it's critical to ask questions about the nature of the cells being sorted. Some example questions might include, are the cells fragile or robust? Are the cells precious and limited in number, or are they abundant and easy to replace? Do the cells have special media or environmental needs? And are the cells labeled with fluorochromes, dyes, or molecular fluorescence? While it may seem like magic, we must keep in frame Shapiro's zeroth law of flow cytometry. There is no magic. Every outcome of our sorting workflow is dictated by close and careful control of both our sample and our sorter. Ultimately, each user will have to dedicate time to determining the pros and cons of each instrument and sample factor that they can control with their desired outcome in mind. In the visual, we see the competing relationship between some key outcomes. Electrostatic sorting will invariably require you to prioritize two of the three and sacrifice the third. Purity is enhanced by well-separated detection, which is greatly influenced by panel design and staining strategy. When paired with a need for recovery, the user must allow for lower speeds and greater time, which can affect cell viability. Media choice and environmental controls quickly become critical. When purity is paired to the need for speed, the user must be willing to accept a significant amount of target cell loss due to coincidence. This use case is typically reserved for sorting into plates or for downstream omics testing. Yield is uniquely enhanced by well-characterized pre-sort viability, as this sets expectations for viable post-sort collection. Instrument operators and SRL personnel will almost always recommend the use of a viability die for both the sort and the pre-sort sample characterization when you count your cells to determine the concentration. A visual field counter like a hemocytometer slide or an automated cell counter with live dead is useful for this purpose. When a need for recovery and speed are paired, the user and operator would likely use an enrichment mode to ensure low or no aborts while accepting that purity will still suffer. This allows the samples to be returned to more favorable environmental conditions quickly. Speed is uniquely enhanced by the ability to select a smaller nozzle size appropriate to your sample paired with a higher frequency. This makes droplets smaller and creates less opportunity for software aborts from cell to cell or intra-droplet coincidence. Smaller droplets created with higher frequencies must also be paired to high-speed electronics, or the advantage of a lowered abort rate may be lost. Small nozzles may also increase the potential for clogs, leading to cell or experimental loss if samples are not well managed and filtered prior to sorting. Because it requires capital and accessory investment, speed is the most costly of the three to influence, as it requires an instrument capable of hosting varying nozzle sizes and fluidic pressure conditions. Some other relevant topics for consideration are included below. The first is biosafety, and while there have not been any reported cases of an operator infected by a sorter, we also know that most operators are not on rigorous biological monitoring protocols. There is a growing concern over aerosol creation, especially related to infectious particles. And since 1997, when ISAC published the recommendations for biosafety, this society has encouraged all operators and users to follow these guidelines to maintain a safe environment for everyone in the proximity of a cell sorter. To this day, no specific federal regulations exist, but there are established rules on certain campuses, most notably on the NIH campus, where there are guidelines for biosafety related to cell sorters. Another topic for consideration is sorter-induced cell stress, often abbreviated as SICS. All electrostatic sorting applies significant pressure and shear stress to cells and exposes them to nutrient-poor environments. While the research continues on this subject, it may be necessary to evaluate metabolic changes caused by sorting if this is an important measurement in your sorting workflow. So what can you as a new user or operator for a cell sorter do to improve your sorts? Well, the first is sample prep. Make sure that you filter your samples as close to their delivery to the sorter as possible. Use an appropriate viability dye that corresponds to your panel design. Concentrate your cells appropriately and verify that concentration with an optical counting method. And make sure to use appropriate sorting buffers that have the right amount of adjuvants and additives to maintain cell viability. As a user talking to an operator, make sure you know thy cells. Be able to describe their characteristics, including their size and suspension. Also, be able to potentially describe the sources of contamination, especially those that would be related to source material or other laboratory environmental conditions. Make sure that when you set up your sample collection, you take pH into account so that the cells can arrive into their sort media with as little stress as possible. And don't forget that while you're sorting, you're also diluting. So you're going to want to start with a high concentration of serum in your collection media. Lastly, be sure to follow established aseptic techniques. Keep your tubes clean and ask your operator to make sure they check the sterility of your sorter's bulk fluidics using a reoccurring Sentinel program. If you've come this far and you want to learn more, I encourage you to start by reviewing the best practices in your flow cytometry analytical methods. Setting gates, adjusting acquisition voltage or gain, doublet discrimination, and thresholding are skills that never go out of style and are always going to aid you in designing robust sorting experiments. If you want to learn even more, you can pursue in-depth cell sorting experimental training focused on the biology of cell sorting, This is often offered locally by your shared resource laboratory or in conjunction with a private instructional company. I would also take this time to encourage you to attend the ISAC CITO meeting held annually every year, typically during the month of May, and to reach out to your local affiliated society to see what training exists as a part of their annual or semi-annual meetings. Lastly, if you're really ready to dive into the deep end, you can pursue instrument-specific operational training. In many SRLs, this is offered by the staff on-site in the laboratory to allow users to become operators and self-support their own sorting experiments. After that, you might want to pursue training delivered specifically by the manufacturer for the instrument that you're using so that you can really hone your skills and become a professional in the art of sorting on the specific instruments available to you. And with that, we welcome any questions and we thank you for your time. I hope that this was useful and we look forward to helping you in your journey in flow cytometry and cell sorting.
Thank you, Matt. Appreciate that very much. We really only have one question that's come in so far. If anybody else has more, please type them into the question and answer box. Our first question has to do with the comparison slide and really microfluidics. The question really is about the output concentration, and this person is commenting that the TITO does have a fairly high concentration of product output, which is very true. I would say that the TITO is probably more unique compared to what I would consider other microfluidic systems, such as the NanoSelect Wolf or the on-chip biotechnology sorter. Those two instruments do tend to put out a product that's fairly dilute. So I agree that the TITO is kind of unique in that bin of microfluidic instruments. I think there are more coming to market soon. So do we have any other questions? Anybody at all? Raise your hand or type it in the box. On behalf of the ISAC community, we would like to thank you for joining our presentation today.
Steve, we got a couple more questions.
Oh, here they come. Good.
So how fast can we go? You speak to the current limits of speed. That's a great question. I think it's really going to be limited by a relationship between the amount of pressure you can put on the sheath stream, and you're pairing that with nozzle size, which is going to be your diameter of your nozzle, and then you're going to pair that again with the frequency, the droplet frequency. So how fast can you create droplets and stably? I know without getting into different manufacturers' claims on this subject, we've seen droplet creation done throughout the history of cell sorting north of 100,000 events per second. That's incredibly fast and requires very, very fast parallel electronics in order to accommodate that kind of speed. The practical limit for speed, though, is also going to be limited by the size of your cells versus the size of the droplet created. So, for example, if you were to take a 100 micron nozzle at, let's say, 30 psi and 40,000 events per second, offhand, you're making a droplet that's about 3 to 3.3 nanoliters in size. If you were going to stack lymphocytes inside of that droplet, you could hypothetically stack somewhere between 3 and 5 end on end if they were touching, right? Obviously, a droplet that big doesn't pair well with the kind of speed that we were describing a moment ago. So I think there's some practical limit, but I would say 15, 20, 25,000 is not completely out of reach, depending on what you're trying to sort and the sorting system that you're using. But this is one of those places where consulting with your SRL staff is going to be critical, because trying to get to those speeds can really affect the cell health and the outcomes of your sorted sample. Tips for exosome sorting. uh, this is an interesting space for research. Not really within the purview of this presentation, but I can touch on it a little bit. Small droplets, obviously small droplets, small tip size, and high frequency are going to be very beneficial for this, even if you're not running them very, very fast. What you want is to reduce the dilution factor associated with the sort. You also need an instrument that's incredibly sensitive. I think it was in 2020, there was a paper published, and I can't remember if it was in JEV or cytometry A. Steve might have to help me out. That was authored by Jennifer Jones, Josh Welsh, and several others, related to sorting on the Astrios. And to do that was an incredible effort. It's not something that you just you start with on day one. So I would say keep looking at the literature. There's a lot of interest in this field right now, but in terms of pragmatic tips, I think that that's very much an in-flight thing that the relevant literature can help answer.
I would add that our manufacturers are working on instruments that are more sensitive and may be able to do this, but we're really not quite there yet. For all practicality, there are a handful of people in the world that have made it work, but just analyzing single exosomes for analysis is challenging enough at this point. So sorting, we're not quite there yet. Do you want to take this next question, Matt?
Can you discuss what parameters to set threshold on only forward scatter A or more recommended? Yeah, so different instruments are going to offer you different thresholding options. A lot of people are going to use forward scatter. Typically, you may see the translation of threshold in your area histogram or bivariate, but it's actually probably thresholding on height. Some instruments have Boolean conditional thresholds where you can stack multiple thresholds. So you can say that a population has to meet multiple characteristics before it can be sorted, and that can be useful. But I would say most people probably threshold on forward scatter if it's appropriate for their cell population. If you're doing something that's really small, like a bacterial sort or a yeast sort, something under 3 microns, your sorter might benefit from thresholding on side scatter and using that as your trigger channel. Steve, do you have anything you'd add there?
No, I think that's a pretty good example. You really have to know which population you're looking at. Backgating is always very beneficial to go back to your forward side scatter and backgate your final gated target population to make sure you know where it's at.
Okay. Kathleen asks, we sort very rare events, CAR-T. Yeah, this comes up a lot, and often find poor viability. Do you have any tips for how to improve this? I would say that you should probably judge your viability in your pre-sort sample as much as you're judging your viability in your post-sort sample, so that you can get an idea of what the impact of the sorting process is on viability changes. This is also where you may want to start looking at a more complicated and moving out of viability and into cell vitality. Looking at cells that are, pre-necrotic or pre-apoptotic, and there's strategies out there that I would leave you to research and define for that purpose. Cells, there's a kind of some colloquial knowledge for people that have run cell sorters a lot, and that's even if you've got a cell that's alive but kind of heading towards death, a cell sorter will help it get there. It will not help cells that are suffering or cells that are on their way to cell death. It will not improve that condition. So it may be really impactful for you to get slices of cell viability or vitality to determine really what your cell health is prior to sorting, and then that would help align your expectations a little bit better. But in terms of pragmatic advice, I would say sort slow, sort with low pressure, You know, those tend to be the things that we try first. Make sure that you're sorting into a median serum rich environment. Make sure that your cells are well separated, and that you're I'm trying to think of some other things. Steve, you got any you want to jump on that?
No, that's pretty good
Yeah.
answer. Sorting is a stressful event, so you really have to keep that in mind. And you know, you've got a lot of material if you're looking for CAR T cells, but you need to go slow and treat them gently if you need to get them back in any viable usage form in the end there. Matt, scroll all the way down to the end. I would really like to hear you, as somebody in industry, discuss what's coming in the next 5 years.
Yeah, I think when you look at the research, you can see where the needs are being created. Obviously, we see movement with imaging technology, the ability to make morphological decisions upstream of a sort becoming hallmarks and new avenues into the science. I think that's really, really important, really incredible. We do see a lot of, I think everyone sees user feedback that would say that being able to design an experiment and analysis and then run that same experiment and sorting is highly desirable. So I think you're going to see more pairwise matching in terms of trying to get sorters and analyzers to detect the same on both systems, which, you know, historically has been something of a challenge, right? They were treated, I think, with a little bit separate discipline in the past. I'm also, frankly, I'm really excited to see where a lot of the microfluidic stuff is headed. I think that parallel processing is a pretty incredible avenue, reducing the volume, increasing the speed, maintaining an aseptic environment, especially as it relates back to anything that would go back into a human, you know, patient. I think are some pretty incredible avenues. And then I also see a growing role, I would say, in clinical uses for sorting. There's some testing that benefits from having a pure population that's been unaltered or unadulterated, and so I see some avenues and some questions there that are going to reveal, you know, that are going to yield some important answers in the coming years.
Okay, I think we're just about out of time, so we'll try and come back and address any of the questions we haven't answered.
Yeah, we may type some of these in.
Okay.
All right.
Very good. Well, thank you, Matt, and thanks everyone for joining us today. If you're interested in other incredible learning opportunities, CytoU does offer a veritable archive of Cyto recordings and on-demand webinars that we've done over the last few years. And we also offer not all of the sessions were recorded, but all of the recorded sessions from Cyto 22 are now available to be viewed online. And please we encourage you to participate in the online survey that you'll get after this webinar. The feedback is very valuable in helping us make things better and maybe even offer some input as to what you'd like to see in the future. And this webinar has been recorded and will be available in a few days on CytoU. So thank you for joining us, and have a great rest of the day. Bye-bye.
Thanks, everyone.